the PowerPoint for tonight because we are going to talk about blood. We're going to talk about red blood cells, white blood cells, different blood diseases, and what exactly does blood do in our system. So blood really has three functions. It transports different things around. It helps to regulate your body, and it also helps protect your body from foreign invaders. So the main thing that is transported in blood is oxygen and different nutrients that go to different cells in the body. But the kidneys, all blood as it goes around the body before it goes back to the heart, the kidneys will filter out blood, and whatever is filtered out of the blood that makes it all the way out of the kidney turns into urine that gets excreted. So our blood also carries different metabolic wastes to be excreted when they come to the kidneys. And when we get to the endocrine system, we will also talk about how hormones are secreted into the blood, and that's how they're transported most of the time to different areas in the body to be used. So blood is also regulates. So it regulates your body temperature. When it gets really warm, more blood vessels towards the surface of the skin actually start to open up more because there's lots of little capillary beds all over in your body, and the capillary systems close it sometimes and open at others, depending on what's going on. And when it's really warm, those capillary beds do warm up. That's why people turn red when they're hot, is because blood is more towards the surface of the body than deeper. Like when it's cold, all these little tiny capillary beds and everything close, and it keeps the blood more towards the center of the body to keep your core warm. It also maintains fluid volume. We need to have a specific level of fluid in the body, and that's blood helps to regulate that because it will get excreted out by the kidneys, filtered out and excreted by the kidneys. If there's too much fluid in your system, your kidneys, your blood takes that to the kidneys to be excreted out. So we don't have just red blood cells, we also have white blood cells. So those white blood cells help prevent infections. We have antibodies, we have complement proteins, and again, those white blood cells. They all help to fight any kind of foreign invaders that come into our system. And we also prevent blood loss through mostly platelets, but those plasma proteins also help to signal more platelets to come to the area to help form clots. We'll look at how some of that stuff works. So, our blood is technically considered a tissue. It's a connective tissue. That's why back in an ANP1, we actually look at blood when we look at different types of tissue because it is considered a connective tissue. We have the cells are living, so they're called formed elements, and they kind of ride around our body in plasma. So blood is made of the term for red blood cells is erythrocytes. That's the anatomical term for them, while white blood cells are called leukocytes, and then platelets are just called platelets. They make that one easy. So, if they were to use a centrifuge, after blood is drawn, A lot of times, to kind of speed up the process, they use a centrifuge to help separate the red blood cells from the plasma because they use plasma to test a lot of different things. So when you use that centrifuge, the red blood cells actually, because they're the heaviest in that tube of everything, they sink to the bottom. It kind of has a yellowish hue to it. So that's about 45% of the blood is those erythrocytes. We have hematocrit, which is the percentage of blood volume that is red blood cells is considered the hematocrit. So in males, their blood is about, or their hematocrit is about 47%, about plus or minus 5% is still considered normal. While females have 42% hematocrit, and it's theirs is could be considered normal. It's still about plus or minus 5% on either side. So white blood cells and platelets, when you separate everything out, here we get what's this little buffy coat in here. Those are where the platelets are. This is this yellow at the top is our plasma. And then the erythrocytes always sit down at the bottom. The erythrocytes in that buffy coat are what's called formed elements because there's actual cells in there versus the plasma that's all liquid. So if you've ever taken chemistry, I am sure you've used a centrifuge before and kind of seen what the results come out. This is what happens when you use a centrifuge with blood. Is everything kind of splits out into three different layers. It's kind of fun to do. So like everything else in the body, the blood has to kind of maintain a specific pH. They the pH for blood is usually, it says the 7.35 to 7.45 is where the blood pH stays, which is pretty much right about in the middle of that pH scale. If you've ever tasted blood, it kind of has a metallic taste because there's iron in it, a lot of iron. And its color is going to vary based off of how much oxygen you have in your system. So if it's if you have a lot of oxygen, it's a very scarlet, bright red. If you have low oxygen, it's a pretty dark, deep red color. So that's one of the things that is always observed whenever blood is drawn, too. Blood technically makes up 8% of your body weight. So one day, if you don't drink enough water, it may affect your body weight the next morning when you first wake up because it's about 8% should be what your blood is, but it's also the waters or the wastes and everything are kind of always being filtered out by those kidneys. So it's kind of, if there's too much water getting filtered out, not enough being replaced, then that can cause some issues down the road if it's a consistent problem. The men tend to have more blood than women. Males have 5 to 6 liters of blood, while women tend to have 4 to 5. So there's little bits of variances just because men are usually taller, bigger than most women out there, so they need more blood to fill more space. Our plasma is almost all water, and it's kind of sticky. That's why if you've ever, I'm sure you have, felt blood when it kind of has a little bit of a sticky feeling to it, and that's because of the plasma. We have all kinds of nutrients, gases, hormones, wastes, proteins, ions, all floating around in that blood because they all have places to go, and the blood travels the most places in the body. So that's why everything goes in the blood. There's plasma proteins that are also in the blood. They and they remain in the blood. There's no cells outside of the blood system that need those plasma proteins. So it's always floating around. And the main plasma protein is actually albumin. It's produced in the liver, and then it is carries molecules around, helps protect certain molecules, and it also contributes to the osmotic pressure of blood, which is another test that's run based off of surface tension on the blood. So here's what is in plasma. Most of the plasma. We have water, we have electrolytes, which are very small amounts of certain cations and anions, positive and negative ions. We have plasma proteins, which are albumin, globulins, gamma, and we have fibrinogen, which is another plasma protein that helps provide the fibrins for blood clots. We also have what are called non-protein nitrogenous substances, such as urea, uric acid, creatinine, and ammonium salts. We have different nutrients that get absorbed in our intestines from the foods we eat, and those different nutrients end up in the blood to get transported other places in the body. There's always respiratory gases, there's always oxygen and carbon dioxide, because oxygen needs to get to all the different cells in the body, and carbon dioxide needs to get removed. And hormones. That's the easiest way for hormones to get from one place in the body to the next, wherever it needs to get delivered, is through the blood and plasma. So, our formed elements of our blood, if you think about that spin tube of blood with everything separated, is made up of red blood cells, white blood cells, and platelets. Red blood cells do not have a nucleus. They do have a nucleus when they are in the process of differentiating into a red blood cell from a stem cell. But once it matures, that nucleus is gone. All other organelles that are found in other cells are gone by the time a red blood cell actually matures. Platelets are actually pieces of a cell. We'll kind of look at platelets a little bit later. White blood cells have a nucleus. Some have two nuclei, some have some weird shaped nuclei depending. Some of them's like a horseshoe, a kidney shape. But most formed elements only survive a few days in the bloodstream. So red blood cells, once they mature, they do not actually divide. They do their job, and then they die off after about 120 days. So red blood cells actually come from the bone marrow, which is found in long bones in your arms and your legs, is where most bone marrow is found. So here's actually what blood looks like under an electron microscope, micrograph. So you can see the 3D structure, you can see the different white blood cells or leukocytes, and then you can see the little pieces of platelets floating around in the blood. And this is what we see in the microscopes in the classroom or in the lab. We see all the little white or red blood cells. We see the different kinds of white blood cells. Then we also see little dots in here that are platelets. So those are the main things that we see when we actually look at blood. So we're going to kind of go over in a little more detail erythrocytes, or those red blood cells, first. Then we'll go over white blood cells, and we'll do platelets. So erythrocytes, or those red blood cells, are very small. So we can only really see each individual cell if we look at it under a microscope. Those red blood cells are actually what carries oxygen around to different cells within the body. They're called the cell is called a biconcave disc shape with it's anucleate. So A means there's no nucleus. And it has no organelles. No, its main function is to transport oxygen and carbon dioxide. So those red blood cells, all they're made up of is hemoglobin, because that hemoglobin is what binds the oxygen and carbon dioxide. Most red blood cells are actually bigger than the smallest capillaries in the body. So as they go through, they go through in almost a single file line as they go through the capillaries. So then we also have plasma membrane proteins that are on some of the erythrocytes and other proteins. So let's have a picture coming up that kind of explains that. So they have a really nice complementary structure for what they do. They have a larger surface area to carry more gas, more oxygen, more carbon dioxide. They're mostly made up of hemoglobin. There's really not much water in a red blood cell. They just kind of float through in more watery type substance. They have no mitochondria, so they do produce anaerobic ATP. If you remember from ANP1 how that was done. So the oxygen that red blood cells carry around, they do not use. They just transport it. They don't use it to create ATP. So they have to use the anaerobic pathway, which only creates 2 ATP per glucose molecule. So here's kind of that structure. We have this nice biconcave shape here. You can kind of see it almost looks like a donut, but it's because you can see where it's concave down in here. Well, the outsides are more raised up. So that helps to transport the oxygen and carbon dioxide around. So since their main job is to transport oxygen and carbon dioxide, that hemoglobin that's a part of those red blood cells binds oxygen, what they say, reversibly. So it binds and it easily lets go of its oxygen when it needs to. So males usually have 13 to 18 grams of hemoglobin per 100 milliliters, and females have 12 to 16 grams of hemoglobin per 100 milliliters. Hemoglobin is kind of this reddish pigment. It's called heme. And it also has a protein globin that kind of those those four globin chains that wrap around each other, kind of like this. So you have alpha globin and beta globin chains that wrap around together. They have the heme group in the middle of each one of those. Each of those heme groups will bind oxygen. This is just the chemical formula of what it looks like. You don't necessarily need to know that for this class. If you go on and take advanced anatomy classes, they may talk about it more. If you go take biochemistry, they'll talk about this more. But I just wanted to kind of introduce it and show it to you. So each hemoglobin molecule, so each of these hemoglobins here can transport four oxygens, four oxygen ions or molecules. And this picture only showed four, but each red blood cell has 250 million hemoglobin molecules. So that's a lot of oxygen that is carried on each red blood cell. But we use oxygen in almost every cell in our bodies, so it's needed. So we get the oxygen when we breathe in. It goes into our lungs, and our heart is pumping blood that's in need of oxygen to the lungs to pick up oxygen. Then it comes back to the heart to go out to the rest of the body. So it produces oxyhemoglobin, which is a very bright red when the blood goes to the lungs. After the blood goes back to the heart to be pumped out to the body, it produces deoxyhemoglobin because that oxygen is being released to the tissues while it's being transported around the body, and it picks up carbon dioxide. So only 20% of the carbon dioxide that's in the blood actually binds to the hemoglobin. So the carbon dioxide that binds to that hemoglobin is actually called carbinohemoglobin because it's got that carbon dioxide, carbaminohemoglobin. If you break it down piece by piece. So red blood cells are produced in the bone marrow, occurs in red bone marrow. And it's formed through hematopoiesis. And all red blood cells and white blood cells, they all come from the same place. And as I said, it's found mostly in the axial skeleton, so your arms and your legs, girdles, so your pelvis and your scapula, and the proximal epiphysis of the femur and the humerus is where most red marrow is found. We have what are called hematopoietic stem cells. And that's what gives rise actually to all the formed elements in the blood. We also have the hormones and growth factors that push the cells towards specific pathways of blood development. So hormones could be saying we need more red blood cells, we need more white blood cells, we need more platelets, we need more of this, more of that. That's what hormones, there's some different hormones actually do. And once the blood is formed, those matured red blood cells that are anucleate, they don't have that nucleus or any organelles in there at all, they enter the blood in sinusoids. to be transported around the body as old red blood cells are taken out of the system. So there's technically 8 stages of that erythropoiesis that makes red blood cells. Those 8 steps or stages takes 15 days. And everything always comes from that hematopoietic stem cell, and that'll transform into myeloid stem cells, which goes to proethroblasts. Those proethroblasts divide multiple times to basophilic etheroblasts. Basophilic etheroblasts go to ribosomes or synthesize the ribosomes. Polychromatic etheroblasts actually synthesize a red hued hemoglobin, which starts to give them their color. And then we get orthochromatic etheroblasts, like, are more pink. This is when at stage 6 is when we lose all the organelles, the nucleus degrades, and as everything is leaving or being degraded within that red blood cell, that's what causes that concave shape. Everything that was holding the middle part of that red blood cell up in more of a cell type shape kind of collapsed down because there was nothing there. We get reticulocytes. We still have some ribosomes at that point. Ribosomes make those proteins. And then we end up with mature erythrocytes, which is what gets transported into the blood. And at that point, any ribosomes that are left are degraded. You do not need to know this whole process. I just wanted to, again, kind of let you know this is the pictorial version of how we start with a stem cell. We go to that proerythroblast, basophilic erythroblast. You can see the nucleus by phase 3, that a nucleus is being ejected out. Everything else is the cells finishing maturing. And what we end up with is the erythrocyte at the end. So there's different things that can happen if we have too many or too few red blood cells. Too few red blood cells actually leads to hypoxia. That's where there's not enough oxygen in the tissues because there's not enough red blood cells to transport oxygen, so the tissues aren't getting what they need. Too many red blood cells increases the blood's viscosity. It gets a lot thicker. So our body is constantly making new red blood cells and constantly getting rid of old red blood cells. So we make over 2 million red blood cells every second. That's a lot. But it's always, of course, because our body likes to stay in homeostasis where it has a balance, and hormones help control to make sure that we're not making too many red blood cells or not enough red blood cells. Also, your dietary requirements, what you're eating versus what you are demanding of your body. That makes a big difference in how many red blood cells are produced. So one of the main hormones that regulates red blood cells is erythropoietin, and it comes from the kidneys because the kidneys are the ones that are constantly filtering that blood every second. There's a lot of blood that's being filtered, so they're kind of monitoring what's coming through and what's leaving. So most of it comes from the kidneys. There is a little bit that comes from the liver. And if there's it'll come from start coming from the liver if we hit that hypoxia where we don't have enough oxygen. So at low oxygen levels, oxygen sensitive enzymes in the kidney cannot degrade hypoxia induced factor. So that can accumulate and it triggers off the synthesis of that erythropoietin to stimulate more red blood cells to be created and released. So, as I said earlier, the causes of hypoxia could be the decreased numbers of red blood cells, could be because of a hemorrhage, so bleeding, could be internal bleeding somewhere, or increased destruction. They're destroying more red blood cells than what are being released. So we haven't, if there's not enough hemoglobin per red blood cell, that causes iron deficiency. We'll go into the different kinds of iron deficiencies. And there could also be reduced amount of oxygen available. If you've ever been to high altitudes and tried to breathe, it gets pretty difficult. If you have some kind of lung problems, have pneumonia, of course, there's fluid in your lungs, so that's blocking the ability of gases to be exchanged when you're in the lung, or when the blood goes into the lungs. So those are two reasons that can cause hypoxia. If we have too many erythrocytes, too many red blood cells, or our oxygen is there's too much oxygen in the body, it slows down the production of erythropoietin. It can so that erythropoietin actually causes the red blood cells to get matured faster. In males, testosterone also enhances that erythropoietin production. So that's another reason why males have more red blood cells. They have the testosterone to produce more red blood cells through that erythropoietin. So as I talked about the homeostasis, our body wants to maintain normal levels for everything. So if there's not enough oxygen getting to the tissues, there could be decreased red blood cells, decreased hemoglobin, decreased oxygen available. The kidneys, sometimes the liver will release that erythropoietin that stimulates the red marrow in the blood or in the bone, or sorry, in the bone marrow to produce more erythrocytes quicker and releases an increased count of red blood cells into the blood so we can carry the oxygen that we need to get to balance ourselves back out again. So our body is constantly fighting to stay in homeostasis. This is just one of the ways that our body is constantly checking what is going on in our system. So there are some athletes that like to use artificial erythropoietin. Because if you think about it, with some of those Olympic style athletes, the more oxygen they have available in their system, they have it helps to increase their stamina. They can go run longer, they can do whatever their sport is, swim longer, and it drastically increases their performance. That's why a lot of those elite athletes actually have to get their blood checked before either they check everybody or they do like spot checks. And they check different athletes to see if there's some kind of abuse of something going on. So if we have too much erythropoietin, if we go from the 45 up to 65, which actually starts to dehydrate the blood concentration. So blood starts to become like sludge because there's not enough plasma in the blood. So blood doesn't move as efficiently, if it can cause strokes because the blood is just kind of slithering its way through blood cells, heart failure because your heart can't pump sludge very well. So it's constantly, this can actually cause problems by using artificial erythropoietin. So to make or for to make red blood cells through erythropoiesis, we need amino acids, lipids, carbohydrates. Amino acids, proteins. Our proteins are made of amino acids. Lipids, those are fats. Your body uses fats to do things in the body. And carbohydrates, we need all of that just to maintain normal red blood cell production. We also need iron from the diet because 65% of iron is found in hemoglobin. So if you've ever had your iron checked in your blood before, a lot of it is actually found. It's why they check it is because it's on the hemoglobin, which is on the red blood cells. It's also found in the liver, the spleen, the bone marrow, and there's also free iron ions but those are toxic. So they tend to bind to different things. Ferritin is like a stored form of iron is what it's known as. That's where iron sits as in ferritin a lot and hemosiderin, and it's transported in the blood by transferrin. This way, there's not free iron sitting around in your blood because that is very toxic to you. B12 and folic acid are very necessary because they help with the DNA synthesis of red blood cells. So we'll go over what a B12 deficiency causes because that actually causes a form of anemia. So we'll get there, but let's look at the lifespan. Red blood cells only last 100 to 120 days. So they're constantly being monitored in the blood through different organs. So since they're anucleate, they do not reproduce. Once they are out in the blood, they kind of, once they're done with their job, they kind of get destroyed. So once they get in the spleen, there's macrophages, which is part of our immune system, that actually help to break down dying red blood cells so that they don't prevent any kind of problems when they are floating around in the blood. Because they could easily break, the hemoglobin starts to degenerate, they can get trapped in the small circulatory channels, the capillary beds that we have. So to prevent that, those macrophages break them down before they can cause problems. So when they are broken down, we end up with heme, iron, and globin. They're all separated apart. That iron will bind to ferritin or hemosiderin. And then they're stored. So I said ferritin is known as a stored form of iron. So that's where the iron from red blood cells that have been destroyed go. Heme is broken down or degraded into a yellow pigment. We also get bilirubin from the liver. That gets secreted into the intestines. And the intestines, as they're breaking down food and breaking down everything else, they break down the bilirubin into urobilinogen. The urobilinogen is kind of like it turns into a brown pigment for that is sterobilinogen, and that's leaves the body as feces. So we get the breakdown of the heme gives us the yellow pigment. Then the breakdown of the urobilinogen and the bilirubin gives us a brown pigment. We get globin is metabolized into amino acids and then released back into circulation to be picked back up to start whatever their next process is. So here's our life cycle of red blood cells. If everything that we've already just talked about, it just gives a visual of the entire process. So there's a lot of erythrocyte or red blood cell disorders. They're either classified as anemia or polycythemia. Anemia is when there's low oxygen carrying capacity. So there's not enough oxygen capacity in the red blood cells for your body to perform your cells and organs to perform their normal functions. We usually see signs of the problem rather than the disease. So fatigue. If you remember what signs versus symptoms. Symptoms is usually what you feel. Signs are what the doctor sees. So you usually feel fatigued, paler. That's when the kind of can be seen because paler is where you're pale, dyspnea, and chills. There's 3 different types of anemia: blood loss, not enough red blood cells, or too many red blood cells being destroyed. So we're going to look at the different anemias that go to each. Blood loss anemias. Hemorrhagic anemia. So if you have some severe wound. So you've lost a lot of blood really fast. This one, luckily, is easy to treat with a blood transplant. So just replacing the blood that was lost can fix hemorrhagic anemia. But there's also chronic hemorrhagic anemia because there's a very slow blood loss occurring all the time. Could be hemorrhoids. Those tend to bleed all the time. A bleeding ulcer, like in your stomach. That's a bleeding ulcer in the stomach is a common chronic hemorrhagic anemia. So finding that ulcer and treating that primary problem will help stop the blood loss, and it can help get rid of that chronic hemorrhagic blood loss or anemia. If we do not have enough red blood cells being produced, is iron deficiency anemia. It can be caused by hemorrhagic anemia, but also by low iron intake or impaired absorption. So we're not getting enough absorption of iron that we need. Red blood cells for iron deficiency anemia are very small. They're called microcytes because they're smaller than that normal size. They're pale. They're not that bright red or deep red color. They're more pale. They can't make the hemoglobin because they don't have oxygen. So the normal treatment for iron deficiency anemia is iron supplements. Iron supplements can tend to upset people's digestive systems. So taking it with food, finding different forms of iron supplements, ones that are slow releasing sometimes makes a big difference versus all at once. All at once can sometimes upset your digestive system. We have pernicious anemia. Pernicious anemia is we need B12. We, the what helps us actually absorb B12 is called intrinsic factor. It's in our digestive tract. So pernicious anemia is actually an autoimmune disease because it destroys the stomach that produces that intrinsic factor. So the intrinsic factor kind of it helps that B12 once it gets into the intestines get absorbed. If we don't have B12, our red blood cells as don't divide from the stem cells to produce more red blood cells because there's not enough red blood cells. They enlarge, get really big, and they cannot divide. So they're called macrocytes. They're usually treated with a B12 injection once a month, usually is what it takes to help get the B12 back into the system. It can also be caused because you're not getting enough B12 in your diet. Vegetarians, it's not as big of a problem for vegetarians nowadays than it was before because they tend to infuse, because they like to infuse foods with things. So they've tended to infuse a lot of different types of things that vegetarians eat with B12 just to try to get enough B12 into their systems too, since it usually comes from proteins and meats. We have renal anemia. It's because there's not enough erythropoietin. It's usually caused by with comes with kidney disease because the kidneys aren't doing their jobs efficiently anyways. So now they're not producing enough erythropoietin. So this is a time when synthetic erythropoietin is actually useful, but it likes to get into the wrong hands of athletes sometimes to try to help themselves. Aplastic anemia is another type because something has happened to the red bone marrow. Drugs, chemicals, radiations, viruses could be unknown. So all formed elements are affected because they all come from the same stem cells and red blood cells or in red bone marrow. So it results in an anemia as well as clotting and immunity defects. So those people tend to get transfusions and they eventually will get transplanted with stem cells to help produce the what's needed to produce the red blood cells, the white blood cells, and the platelets again. We also have where we said there's too many red blood cells being destroyed. And if there's too many red blood cells being destroyed, we tend to get that premature lysis or the destruction of those red blood cells. And that's known as hemolytic anemia. If you think of lytic as they're breaking them down, lysis, lytic, it's the same thing. So it could be the cause. I know I talked to some people on Tuesday about incompatible transfusions. So you get a blood transfusion that's just not compatible you, with your blood that you have. It can happen because of hemoglobin abnormalities. It's usually some kind of genetic disorder. And those are either called thalassemias or sickle cell anemia. I'm sure most of you have heard of at least sickle cell anemia. Let's look at thalassemias first. So, a thalassemia is usually found in people of Mediterranean ancestry. So, one of those globulin chains causes a problem because it's not there or it is faulty. They can't do anything with it. So, the red blood cells are very thin, delicate. They don't have the hemoglobin that they need. And there's many different subtypes. So, they could be really extreme and severe. It could be very mild. And if the very severe cases end up getting monthly transfusions just so they can carry on with their daily life. Well, sickle cell anemia is actually a mutation in the hemoglobin. And as it says, there's only one amino acid that's wrong in that DNA chain that creates the red blood cells. There's 146 amino acids in hemoglobin or in that globin chain, and only one of them is wrong, and it causes sickle cell anemia. They become like a crescent moon type shape when the oxygen levels are too low. So during exercise is a big one. And they get stuck very easily in those really small capillary beds, really small blood vessels. So we're not getting enough oxygen that we delivered as we need, and it can cause extra pain. It is more common in Black people of the African malarial belt and their descendants. This is where majority of cases have been found. There's one possible benefit. They don't contract malaria. And malaria is pretty common over in Africa. So it actually prevents them from getting malaria. So I mean, it's not a good to have sickle cell anemia, but there is one benefit to it because sickle cell kills about 1 million people a year. It takes a special kind of hemoglobin to cause sickle cell. They need 2 copies of the hemoglobin S. That's what it's called. And individuals with only 1 copy of that hemoglobin S will have either a milder chance or they have a much better chance of surviving malaria as well. Treatment, they usually do some kind of transfusions during crisis. They can inhale nitric oxide and they try to prevent that sickling. So they use hydroxyurea during the formation of fetal hemoglobin to cause it to not sickle. They've done stem cell transplants. They've used gene therapy. And they've used nitric oxide as well, all trying to prevent the sickling of the blood cells. So here's what it looks like in an electron micrograph. Here's a normal red blood cell here versus this is what happens when that blood cell sickles. So as you can see, it can pretty easily get stuck in different areas because it's almost got like little hooks on the top or the bottom that can get it stuck in those really small capillaries. Polycythemia. That's what it's called when we have too many red blood cells. It's usually for an abnormal reason. But again, that blood, as we talked about earlier gets kind of sluggish, so it doesn't move as easily. There's two there's two different kinds. There's polycythemia vera, which is marrow cancer that creates too many red blood cells, which causes the hematocrit to go as high as 80%. So they tend to do therapeutic phlebotomy. They cannot donate blood because there's so many red blood cells, but they take out blood to try to thin out the excess amount of red blood cells. So they usually have to go to a hospital to do it, and the blood gets destroyed because there's nothing they can really do with it. There's also secondary polycythemia. Secondary, anything that says secondary means that there's another something else that's causing that condition. So secondary polycythemia can be caused by low oxygen levels at high altitude or too much erythropoietin production. So if that low oxygen at high altitude, you come down off that high altitude, that would fix that secondary polycythemia. Increase erythropoietin production. There's ways to calm that down or quiet it down so the production isn't as abnormal. Blood doping. This is something else that athletes do. They remove some of their blood, they store it, they reinfuse the red blood cells in before an event. This is another, it's kind of like a way of that artificial erythropoietin, but they're not really adding more chemicals to their body. They're taking out blood. Your body reproduces what you took out. Because if you donate blood, they usually take a liter of blood from you. And that liter of blood, your body replaces. So, right before an event, these athletes put the blood back in themselves, inject the blood back in, because that gives increased oxygen. So now they can, whatever event they're trying to do, they have more stamina. They can go for longer. So that's kind of their way of not necessarily using artificial, but they're considered blood doping. They're using their own blood. Let's look at leukocytes or white blood cells. They are the only formed element that's considered a complete cell because they have a nucleus, they have the organelles still remaining after they're fully mature. So white blood cells are about 1% of the blood volume. So there's usually 4,800 to 10,800 per microliter of blood. White blood cells' main job is to fight diseases. As they travel around the bloodstream, they can actually leave the capillaries by what's called diapedesis. It's where they kind of slither in between the cells that are holding those capillaries together and go out into outside of the capillary beds into the tissues to do whatever they might need to do. So they move through those tissues in an ambiboid motion. So they kind of just slither their way around, and they use positive chemotaxis to get to whatever may be invading the body. So leukocytosis is if we have too many red blood or white blood cells. So we have over 11,000 per microliter. And it's most of the time when that happens is because your body's fighting some kind of infection. Cold, virus, Those are the two main reasons why you'd end up with leukocytosis, is you're fighting some kind of infection. There's two different kinds of leukocytes, or two different categories, I should say. There's what are called granulocytes and agranulocytes. Granulocytes, they contain granules. When they're dyed, you can see individual granules in there. And that is neutrophils, eosinophils, and basophils. Agranulocytes do not have visible granules, and those are lymphocytes and monocytes. The mnemonic to try to remember the different kinds of red blood cells is never let monkeys eat bananas. Never neutrophils let lymphocytes, monkeys, monocytes eat eosinophils, bananas, basophils. Those are actually in decreasing abundance order. So that tells you neutrophils are the most abundant white blood cell, while basophils are the least abundant white blood cell. So if we see, out of all the white blood cells, neutrophils make up about 50 to 60%. Lymphocytes make up 25 to 45%. Then we get monocytes, 3 to 8%, eosinophils, 2 to 4%, and basophils is 0.5 to 1% of all the white blood cells. So we'll go over what each of those white blood cells does. But if we look, a neutrophil is, this nucleus is considered multi-lobed because they do connect to each other. So it may look like there's more than one nucleus, but it has connections here between each parts. So it is considered multi-lobed nucleus. It stains like a red and blue. And you can see in the cytoplasm out here, all these little speckles or dots, those are all granules. The eosinophil has what they call a bilobed nucleus. So there's this one and this one, they're kind of connected in the middle. And they have lots of red granules. There's tons of little red granules in here. They all stain red in an eosinophil. So the neutrophils are more of like they almost look purple to me, like purple granules, while eosinophils have red granules. So that's one way to kind of tell them apart. The neutrophils also have multi-lobe nucleus. Like this top one has several lobes technically. This bottom one, this is a lobe, this is a lobe. This could possibly be a lobe. It looks like they're kind of starting to separate apart. So it's considered multi-lobed, while eosinophils have a bilobe, so there's only two. A basophil, you can't really see the nucleus. All you see are these like purplish black granules. This is the key characteristic of a basophil, is you really cannot see the nucleus in it because the granules stain so dark. Now the agranulocytes, these lymphocytes, the nucleus is almost as big as the entire white blood cell on a lymphocyte. So that nucleus is purple-ish, while it's a lighter purple on the outside. And a monocyte, it says it has a kidney-shaped uterus. It kind of, or nucleus, it kind of does. But if you compare the nucleus of the monocyte to the neutrophil and the eosinophil, the two granulocyte ones that have more of a bilobed or multi-lobed nucleus, you see how they're separated versus the monocyte is just one big nucleus in a kidney shape. So it's one of the ways to kind of tell each of those apart from each other. So granulocytes are those neutrophils, eosinophils, and basophils. Their lifespan is a lot shorter than red blood cells. They're also a lot bigger than red blood cells. So as I said, they have a lobed nucleus. The type of stain that they use is called a Wright's stain. You don't need to know that. Just if you ever work in a lab where you're dyeing, using dye to find things, that's just the dye that you would use. All three granulocytes are phagocytic to some degree. So neutrophils, again, they're 50 to 70% of the white blood cells. They're twice as big as those red blood cells. And the stains are both acidic and basic because the different dyes they use as an acidic dye and then a basic dye to get the colors. They have hydrolytic enzymes or antimicrobial proteins that are called defensins. Because of their nucleus is lobular, they can have anywhere from 3 to 6 lobes. They specifically, neutrophils are known for killing bacteria. They kill microbes through what's called a respiratory burst. So they synthesize an oxidizing substance. Yes, white blood cells synthesize almost something like a bleach or hydrogen peroxide. And these defensins, those hydrolytic enzymes or antimicrobial proteins, come together with this oxidizing substance and they phagocyze bacteria. So they kind of ingest the bacteria. So here again is that neutrophil. You can see the different lobes. You can see like the purplish granules in the cytoplasm. Eosinophils. Those are about 2 to 4% of all the leukocytes. Their nucleus is always bilobed, so they always have 2 lobes. I like this one. Your book says that it resembles earmuffs in an eosinophil. If something like that helps you remember the eosinophil versus something else, it's whatever works. Sometimes it's something weird and strange or silly that helps you remember something. So eosinophils go after parasites. They also contribute to allergies and asthma and the immune response. But they're mainly known for parasites. So here is that bilobe nucleus. If it looks like earmuffs, great. If not, then but you can find something else that helps remember those. Basophils. Basophils have that deep purple. You can't really even see the nucleus. They're also the rarest type of white blood cell, 0.5% to 1%. They contain histamine. Histamine is a type of inflammatory chemical in the body, and it helps to bring white blood cells to that inflamed area. So if you have a lot of histamine in your skin, and like it causes red very easily on your skin, like a reddish hue to the skin tone. That's because of histamine. And basophils function similarly to mast cells. If you've never taken microbiology, that is usually where they talk about mast cells. So mast cells, they also go after infections. They're involved in, again, those immune reactions. They're usually found all over our system. So they're basophils and mast cells are very similar. So here's that basophil. All you kind of see are those blackish granules with some purple mixed in there. And that's kind of what makes them different. We have our agranulocytes. They don't, when they stain, they really don't stain granules like the granulocytes do. So we have lymphocytes and monocytes. We look at our lymphocytes, they're about 25% of the white blood cells. They have a dark purple nuclei with a bluish cytoplasm. They are mostly found in lymphoid tissue. So they kind of tell you lymphocytes, lymph, where they're at. You'll find some in the blood, but not as common as in the lymphatic tissue and the lymph nodes, the spleen. We have two different kinds. We have T cells or T lymphocytes and B cells or B lymphocytes. T lymphocytes go after viruses and tumors. B lymphocytes help to produce antibodies that'll fight different viruses and bacteria. So here's our lymphocyte. It has a very large nucleus that kind of takes up most of the cell. And it's very purple versus the lighter purple in the cytoplasm. They say it's a pale blue, but this picture kind of looks more purple to me, at least. The other agranulocyte is a monocyte. They're the largest of all leukocytes. There's 3 to 8% in the white blood cells. They have a U or a kidney-shaped nucleus. They are phagocytic cells, so they fight viruses as well, bacterial parasites, and chronic infections. So active lymphocytes, or they activate the lymphocytes to mount some kind of immune response to get your whole immune system fighting against whatever could be invading your system. So here's that monocyte. It's got that very large kidney-shaped nucleus with a lighter purple, or they still say it's a blue cytoplasm. So here's again all the different leukocytes or white blood cells to tell them all apart. Try to figure out which one is which. So leukopoiesis is the creation of white blood cells. And there's two different chemicals that stimulate the growth of, or the production of white blood cells: interleukins and colony stimulating factors. Interleukins are numbered. There's different kinds that produce different things, different types of leukocytes. And colony stimulating factors, they're named for the white blood cells they stimulate. So granulocyte colony stimulating factors stimulate the granulocytes. Agranulocyte stimulating or colony stimulating factor stimulates the agranulocytes. So all leukocytes originate from hemocytoblast stem cells, and they branch into different pathways. The lymphoid stem cells for lymphocytes, or the myeloid stem cells for all other elements. So here's the steps to production of the granulocytes. You don't need to know each individual step. It's not worth the time. So we have myeloblasts that come from the myeloid stem cells, line of stem cells. They are promyelinate. They go to promyelocytes that accumulate lysosomes, and they go to myelocytes that accumulate granules, to band cells to form a nice curve to the arc. Then they mature to granulocytes. Their nuclei becomes segmented before it's released into the blood. And there's 10 times more stored in the bone marrow than in the blood. We have 3 times more white blood cells are formed than red blood cells because white blood cells have that shorter lifespan. It's cut short because they're constantly fighting. There's always something invading your system. It's just how your body reacts to fight it off, to fight back. So a lot of time you don't know that something's invaded your system because your immune system fights it off before you ever feel any effects from it. Agranulocytes. We have monocytes that come from the myeloid line, and lymphocytes come from the lymphoid line. You don't need to know these steps either, but monocytes can actually live for several months, while lymphocytes surprisingly can live for a few hours to decades, just depending on what they could be used for. And here's the different formations of the different white blood cells, how they get from their stem cells to their mature white blood cells that get released into the system. So many hemopoietic hormones, like erythropoietin and colony stimulating factors, are used clinically. They can be used to stimulate bone marrow of cancer patients receiving chemotherapy or stem cell transplants. They can also be used to increase the protective immune response of AIDS patients. So these could help keep people who are already fighting something else off healthier than what they would be otherwise. So there's different leukocyte disorders as well. We can have overproduction of white blood cells or abnormally low white blood cell counts. So if we have too many white blood cells, it's known as leukemias and infectious mononucleosis. Or we can have abnormally low white blood cell counts, which is leukopenia. Can be drug induced. It's usually cancer drugs that cause that. Glucocorticoids can too, but it's more commonly seen in cancer patients. So leukemias are a type of cancer where we have the overproduction of abnormal white blood cells. And it starts with the clone of a single abnormal cell, and that single abnormal cell keeps reproducing over and over and over again. So they're named based off of that abnormal white blood cell clone that creates the problem. So we get myeloid leukemias and lymphocytic anemias or leukemias. Myeloid involves the myeloid descendants, while lymphocytic leukemias involve lymphocytes. Acute leukemias derive from stem cells. That's common in children, but there's also chronic anemia or leukemia. Sorry, I keep saying that. Involves the proliferation of later cell stages. So that's more common in elderly. All leukemias are fatal if they're not treated. So we end up with immature, non-functional white blood cells in the bloodstream. There's nothing, they can't perform their jobs they're created for because they're immature, so they can't do their job yet. So cancer cells end up filling the red bone marrow and crowding out other cell lines. So when you have leukemias, anemia is usually involved. Some forms of bleeding are involved. People usually end up dying from an internal hemorrhage, which is internal bleeding, or an overwhelming infection because the body can't, since the white blood cells aren't doing their job of fighting infections, you tend to get sick a lot easier. Takes a lot longer to fight off different infections. So irradiation, trying to get rid of that stem cell that is creating all the incorrect white blood cells. We have our anti-leukemic drugs and stem cell transplants, all for different kinds of treatments. We also have infectious mononucleosis. It's known as the kissing disease. It's very common in young adults. I had a friend in high school who had it, and it took her over a month to get rid of it. It's was caused by the it is caused by the Epstein-Barr virus. So that virus results in high numbers of agranulocytes. So it usually involves those lymphocytes, and they become enlarged. Lymphocytes aren't that big. These become a lot larger. And they originally thought that stem cells, or they thought the cells were monocytes. So the disease is named mononucleosis. That's where the name came from, because monocytes, mononucleosis, it's not what it is. They're involved in lymphocytes. You get tired, achy, a chronic sore throat, a low fever, and it takes 4 to 6 weeks, a month to a month and a half to feel better. Platelets. Our platelets come from megakaryocytes. They are actually fragments that break off of those megakaryocytes. And when you look at blood under a microscope that has been stained, they stain purple. They have these little purple granules throughout, and those are all platelets. They actually have serotonin, calcium, enzymes, ADP. ADP is not the usable form of energy. It's adenosine diphosphate, and also has platelet derived growth factor. And all of that is actually used in the process of clotting blood. So those usually follow platelets around. So they form temporary plugs in blood vessels, is what their job is with platelets. They end up clumping together to clog whatever hole or what could have caused any kind of hole or anything in the blood vessel to prevent blood from continuously leaking out. So platelets, when they're circulating in the blood, are not active. They are actually mobile by nitric oxide and prostacyclin from the endothelial cells that line blood vessels. That's how they kind of get around to where they need to go. Thrombopoietin is what regulates the formation of platelets. And again, they are formed from the myeloid line of megakaryocytes, which form those megakaryocytes. And then the platelets are actually break off of the megakaryocytes and start moving around in the blood. They only last 10 days. It's not very long before they're cleaned out of the blood as new ones keep being replaced constantly. So platelets usually have 150,000 to 400,000 per milliliter of blood. It's a lot of platelets. So here's the stem cell, the hemopoietic stem cell that all formed elements of blood come from. It turns into a megakaryoblast, which becomes a megakaryocyte. By the stage 4 megakaryocyte, these little pieces that are breaking off of this megakaryocyte are forming platelets. This is how platelets are formed. They literally just break pieces just continuously break off of it. And that's what makes platelets that are moving around in our blood. So here's a chart for you to use that just shows all the different formed elements in the blood. It describes them, says about how many types of cells are found per microliter of blood, how long they take to develop, and their average lifespan, and what do they do. I think the only thing for the white blood cells that I ask is anything about could be what their functions could be. So knowing, oh, neutrophils, those go for bacteria. Eosinophils, parasites and allergic reactions. Because if you're ever looking at blood results, if one of those white blood cells is elevated, you can tell what kind of infection somebody is fighting. Oh, they have a bacterial infection. Oh, they're fighting a virus. They have a parasite. That's why there's an increased amount of this certain type of white blood cell. So those are the only things that I ask for the white blood cells, just because it helps with reading blood results. And then the red blood cells, pretty sure I asked something about this, how many, and about how long their lifespan is. Hemostasis. This is the process of stopping bleeding after something has poked a hole through a blood vessel. So there's different clotting factors and substances that are released by platelets when that tissue is injured to bring more platelets to the area. So the first step is there's some kind of vascular spasm. So that vessel responds to that injury. They constrict. So they constrict, they get smaller to try to help prevent bleeding. The direct injury to the vascular smooth muscle. Blood cells or blood vessels have smooth muscle, so that's not something we can really control. There's chemicals that are released by those endothelial cells in that blood vessel. We also get platelets that are released. We have pain reflexes. That's why sometimes when you get poked by a needle, sometimes it hurts. The smaller the blood vessel, the more effective this process is, because there's not as much space to close down. So they're trying to prevent the blood flow to prevent blood loss while that blood vessel gets repaired. The second step is a platelet plug formation. So platelets start to stick to collagen fibers from the blood vessel that kind of sticks out once that blood vessel's been poked through or whatever happens. Platelets cannot stick to the blood vessel walls because platelets stick to collagen. They don't stick the walls, the endothelial cells. If platelets stuck to those endothelial walls would cause all kinds of problems. We need our blood vessels walls to be smooth so things pass through easily. So we also need prostacyclins and nitric oxide, which is secreted by the endothelial cells that line the blood vessels. Those actually act as well to prevent the platelets from sticking. There's von Willebrand factor that helps stabilize platelet collagen adhesion. So once the blood vessel's been punctured, collagen is available for the platelets to start sticking. When that blood vessel constricts, it helps to activate the platelets to swell as well, to get bigger. And they release chemical messengers, those platelets do. ADP actually causes that adenosine diphosphate causes more platelets to stick and release their contents. We use serotonin and thromboxane A2 to enhance that vascular spasm even more. So that blood vessels pushing down even more, so those platelets stick together even more. This is, if you remember back from Anatomy 1, this is actually a positive feedback cycle. Positive feedback is when it enhances something. So it makes whatever process is happening happen even more. So we're sending chemicals out to bring more platelets to the area. As platelets get there, they release more chemicals to bring more platelets. So we're bringing more and more. So that's what makes it a positive feedback cycle. Platelet plugs are good for small vessel tears, but if a larger blood vessel tears, that's usually where something like stitches come into place, because it's hard to close down an entire blood vessel, a large blood vessel, to form that platelet plug. Once that platelet plug comes into place, we get blood clotting. That blood that clots to that area reinforces that platelet plug, helps keep it in place. The blood is transformed into like a liquid or a gel to help reinforce. Clotting factors are used, plasma proteins are used, all to create this platelet plug down here. And if it's right at the surface of the skin, that's where the scabs form. If it's not at the surface of the skin, you don't see this whole process happening in a blood vessel. You only would see it if it's more at the surface. So in coagulation, there's two pathways for the prothrombin activator. There's an intrinsic and an extrinsic pathway. They use both because it's triggered by tissue damaging events. So something damaged that tissue of that blood vessel, and it involves a series of procoagulants. So each pathway cascades towards and ends with the activation of factor X. With factor X, it complexes with calcium, platelet factor 3, and platelet factor 4 to form prothrombin activator. So it's what helps keep that platelet plug in place until it's no longer needed. So that intrinsic pathway is called intrinsic because clotting factors are present within the blood. So it's triggered by negatively charged surfaces, such as those platelets and collagen coming together, or even glass or a test tube. The extrinsic pathway is because things are coming from outside the blood to help form that clot, such as that tissue factor or or factor 3. And this helps to bypass steps of the intrinsic pathway so that extrinsic pathway is actually faster to form that platelet or that platelet plug and the blocking off of the blood vessel. So this chart just kind of shows both pathways, intrinsic and extrinsic, and how the extrinsic pathway is so much quicker. So in phase 2, get our pathway to thrombin. Thrombin or prothrombin activator then catalyzes the transformation of prothrombin to thrombin, which helps with the plug to the clot to the blood vessel. And then phase 3 is the fibrin mesh. So we have fibrinogen in our blood that goes to fibrin. The fibrin strands, if you remember, those are from the tissue chapter way back in chapter 4. They form the basis of that clot. Fibrin causes a plasma to become a gel-like structure, which causes the fibrins to kind of cross-link, and it strengthens and stabilizes that clot. Keeps everything formed how it should until it's no longer needed. We also have anticoagulants, which are factors that normally dominate in blood and inhibit coagulation. So some people that have their blood clots way too easily can be given anticoagulants because their blood clots are causing more harm by random clotting where it shouldn't happen than when it should be used, such as when there's some kind of puncture to a blood vessel. So when we have that blood clot, we have that fibrinogen that comes in and forms fibrin, which helps to make a fibrin mesh through that clot. So if we look, here's that whole pathway again. It shows the extrinsic and intrinsic and how they come together, and how the extrinsic pathway again just skips lots of steps. Here is that fibrin mesh in an electron micrograph. This is what a clot actually looks like. It kind of looks cool. So these are just the different blood clotting factors, the different things that help and where they come from, what they actually are. Most of them are some kind of plasma protein. This is just kind of for your information. I'm not going to ask you what specific factor does what. That's way too nitpicky. So once the blood vessel has been repaired, we have a process called clot retraction. They have to remove that clot. Just like in skeletal muscle, if you remember the actin and myosin, there's also actin and myosin in the platelets that, that contract within 30 to 60 minutes. The contraction pulls those fibrin strands, squeezing serum from the clot. Serum is plasma minus the clotting proteins. It draws the ruptured blood vessel ends together. So it helps kind of pull everything together. So both damaged ends end up coming together. The vessel or the blood vessel is healed even as the clot retraction occurs. So the platelet derived growth factor is released by platelets. It stimulates the division of smooth muscle and the fibroblasts to rebuild that vessel wall. Then the vascular endothelial growth factor stimulates endothelial cells to multiply and restore endothelial lining. So the cells that are already there start to reproduce to fill in where the damage had occurred. We end up with fibrinolysis. It's where the clots are finally removed after everything is repaired. So that usually begins 2 days, but it takes several days for that clot to finally be dissolved. So plasminogen, which is a plasma protein trapped in the clot, ends up being converted to plasmin, a fibrin digesting enzyme. We also have tissue plasminogen activator, or factor 11, and thrombin all play a role in that conversion process. There's different factors that limit normal clot growth. We don't want those clots to get too big. So we also want them dissolved completely. We don't want them being broken off and released into the bloodstream because that is what can cause stroke. So there's two mechanisms that help to limit that clot size: the swift removal and dilution of clotting factors. And we also have the inhibition of activated clotting factors. So the limited amount of thrombin is restricted to clot by fibrin threads, and it prevents the clot from getting too big or escaping into the bloodstream. We get antithrombin 3, helps inactivate and any unbound thrombin that escapes into the bloodstream. Heparin is basophil and mast cells inhibit that thrombin by enhancing antithrombin 3. So heparin is actually a drug that is given to help prevent clotting, because some people's blood clots more than others, and that's a way to kind of help prevent strokes. So there's different ways to prevent undesirable clotting. We don't need clotting unless there's some kind of damage to a blood vessel. We have a smooth endothelium of blood vessels, helps to prevent platelets from clinging together. So that's the inside of the blood vessels. They're nice and smooth. And those endothelial cells that line the inside of those blood vessels will secrete nitric oxide and prostacyclin. That helps prevent things from sticking. Vitamin E quinone is formed when vitamin E reacts with oxygen, which helps prevent any clotting. It's an anticoagulant. So these are different ways to keep blood vessel or clots from forming when they're not needed. different types of disorders. We get thromboembolic disorder. It's where clots form when they don't need them. We also have bleeding disorders where normal clots are not forming. They're both disorders are disseminated intravascular coagulation is what they are called. So thromboembolic conditions, a thrombus is a clot that develops and it stays in an unbroken blood vessel. So it can block circulation and it'll eventually lead to the death of the tissues in that area because they're not getting the nutrients that they need. We also have an embolus, which is that thrombus that broke off. Now it's free floating in the blood. That's not good. That's where problems start to really occur. And an embolism is where that embolus that was freely floating in the blood obstructs a blood vessel. Pulmonary, it happens in the lung somewhere, or cerebral, happens in the brain somewhere. Happening in the brain somewhere, that's where a cerebral emboli is usually what causes a stroke. Because your brain's not getting the blood supply that it needs to function correctly. So people who have atherosclerosis, which is a buildup of cholesterol and other things in the blood vessels, too much inflammation in the body can cause this. If your blood is not, if it's flowing too slowly, or blood stasis, so it's not moving. Stasis means there's no mobility, it's motility, it's not moving. Those are specific risk factors for having a thromboembolic type of condition. Then there's anticoagulant drugs. These are used for preventing clots. Aspirin is like low dose aspirin is used a lot, but it's not something you should take without it being prescribed by your physician. Don't just take it just to take it, because then it's not healthy for you if you don't really need it. Aspirin is an antiprostaglandin and inhibits thromboxane A2. So a lot of times elderly take it because it lowers the risk of a heart attack by up to 50%. That's a pretty significant amount. There's also heparin, which I kind of mentioned before. It's used pre and post operative for cardiac care, and it prevents venous thrombosis. So getting those clots forming and sticking in veins somewhere. Warfarin is another anticoagulant to help reduce the risk of stroke patients. And it's mostly used for people who have some kind of atrial fibrillation, so their heart's not pumping the way it should. So blood tends to pool in the heart. Warfarin interferes with vitamin K, the action of vitamin K in the body. So if people are on warfarin, they do also need to take vitamin K so they can still get the benefits of vitamin K as well. Thrombocytopenia. Anything that is penia, and it refers to anything, penia, means it's not enough. Too slow, there's not enough number, of platelets circulating around and thrombocytopenia. But penia means that there's not enough of something, it's below normal. So they end up with petechiae, or that kind of pop up all over everywhere, and they get hemorrhaging, which is internal bleeding. So it's usually due to the suppression of the red bone marrow or destruction. Again, malignancy, so some kind of cancers, radiation, or drugs. So if the platelet count is less than 50,000, that is diagnostic. That's where people get diagnosed with thrombocytopenia, and they start looking at transfusion of platelets specifically, so that people don't end up with hemorrhaging or internal bleeding in the body. So here's what the petechiae looks like. They're little red spots all over the body. This is from somebody who has the thrombocytopenia. Can also have impaired liver function, because your liver synthesizes the procoagulants or the clotting factors. And if your liver's not producing those procoagulants or clotting factors, that can cause some bleeding disorders. Because if we don't have enough vitamin K, this is where if people are on something like warfarin, they need to be taking vitamin K, because it can cause bleeding disorders. You can get from hepatitis, cirrhosis, which involves the liver. Liver disease can also prevent liver from producing bile, which is needed to absorb fat and vitamin K. So liver disease can cause all kinds of problems. And cirrhosis is usually caused by heavy drinking for many, many years. So it's pretty common in alcoholics. Hemophilia. They're hereditary bleeding disorders. There's 3 different kinds. The most common is hemophilia A, and it is due to factor 8 deficiency. Hemophilia B is factor 9 deficiency, and hemophilia C is factor 11 deficiency. I'm not going to ask you what each of those specifically are. That's, again, too nitpicky. So they usually have prolonged bleeding, so it takes a lot longer to clot, and they can have bleeding into their joint cavities, which causes joint swelling. So treatment is usually injections of the different factors, genetically engineered factors, which has actually reduced and eliminated the need for plasma transfusions. So they've learned how to genetically engineer ways to help people with hemophilia, which is kind of cool. There's disseminated intravascular coagulation. That involves both clotting and severe bleeding. So they have both ends of the spectrum kind of going at the same time. They have widespread clotting that occurs in intact blood vessels, so that blocks the flow to certain blood vessels. And then they also have bleeding that causes residual blood to not be able to clot because clotting factors are depleted. This can occur in what's called septicemia, incompatible blood transfusions, and complications in pregnancy. let's look at blood transfusions. We did with blood typing that we did on Tuesday. The reason why this can be important is for people who work in hospitals where people are getting blood transfusions. You may be told this person has this blood type, and then you need to go get blood for somebody. So you need to know what kind of blood works for them and what kind does not. So our cardiovascular system tries to minimize the effects of blood loss by reducing the volume of affected blood vessels. So our when we get to the cardiovascular system, we'll look into that more, but our blood vessels can constrict and send blood different directions. It can also open up certain blood vessels to let blood go to certain tissues when needed. And they also, that cardiovascular systems can step up the production of red blood cells. So our body can compensate for only so much blood loss. 15 to 30% causes pallor and weakness. Pallor is the paleness of the skin because there's not enough blood going to the layers underneath your skin to keep the skin tone you have. Weakness because there's not enough oxygen going around to your system. If we get more than 30% of our blood lost, your body could end up in shock, and it could be severe enough that it could be fatal, which is not a good thing. So death from shock usually ends up occurring because of low blood pressure. If you don't have enough blood to pump around, your blood pressure is not as strong, and it lowers your blood pressure down because there's not as much blood circulating in the system. So to prevent death from shock, volume has to be replaced. So they can use saline or a multiple electrolyte solution, which is called Ringer's solution. And it helps to minimize the plasma electrolytes. That helps to restore blood pressure by having enough volume in your blood vessels. Replacement of that volume restores adequate enough circulation, but it doesn't replace the oxygen capacity of the red blood cells, unfortunately. So they end up using whole blood transfusions as well. But that's only when blood loss is rapid and substantial. So there's a lot of blood loss and it happens very fast. They can infuse packed red blood cells, or as they call PRECs. Plasma and white blood cells are removed. And if packed red blood cells are actually used to restore oxygen carrying capacity. Blood banks usually separate the donated blood into components because blood has a shelf life of about 35 days. Human blood groups of donated blood must be determined because different transfusion reactions can be fatal. So we looked into these blood types Tuesday, and our red blood cell membranes have different antigens on them. An antigen is anything perceived as foreign that can generate an immune response. So red blood cell antigens are referred to as anti-agglutinogens because they promote agglutination. A mismatched blood or transfused blood is perceived as foreign and may be agglutinated and destroyed. So it could be very fatal. Humans have at least 30 naturally occurring red blood cell antigens. There's could be the presence or absence of each antigen to help classify the different groups, which is what we did in lab on Tuesday. There's other blood groups, which are some of them are MNS, Duffy, Kell, and Lewis, but they're pretty weak. So they're not usually typed because they don't make as big as of a they don't create as big of a problem in most people. It may if somebody needs several transfusions, they'll start looking into that. But if they only need one or two transfusions, it's not going to be that big of a deal. So the main antigens that are looked for was we have the ABO and the RH blood groups. Those are where most of the problems will occur if they're not matched up correctly. So type A blood only has a agglutinogen. Type B has only that B agglutinogen. AB have both A and B, while type O has neither. So blood can contain performed anti-A or anti-B antibodies. They act against the transfused red blood cells of the ABO antigens, not present in the recipient's red blood cells. So anti-A or anti-B form in the blood about 2 months of age. And by 8 to 10 years of age, they're at adult levels. So by 8 to 10 years of age, you really have to match up the blood type. So we have AB blood type. It shows different, it shows spikes for the A antigens and circles for the B antigens. And this chart also shows what percentage of the population of the different races have these types of blood cells. I thought that was just kind of interesting. That's not something you need to know, but ABO is the least common blood type because they have both A and B antigens. They are considered a universal recipient. They can receive A, B, AB, and O blood type. They can receive blood type from every type. While B has those B antigens and they contain anti-A agglutinins. They can receive B and O, while A has those A antibodies and the anti-B antibodies. They can receive A and O blood type O. They are that universal donor. You notice O is in every single category, so they can give blood to everybody. But they can only receive O blood type. But if you look, most of the population in this world seems to have O blood type. Or a large percentage of it anyways. A is the next most common. So O can give to everybody, while AB can receive from everybody. That's something important to know. Now if we look at Rh factors, there's technically 52 named Rh agglutinogens. C, D, and E are the most common types. We just look at them as that Rh factor as Rh positive or Rh negative. So Rh positive technically indicates the presence of D antigens. You don't need to know that specifically. Just know that most of the population is actually Rh positive. While anti-Rh antibodies are not spontaneously formed on Rh negative individuals, but anti-Rh antibodies form if an Rh negative individual receives Rh positive blood, or if an Rh negative mom is carrying an Rh positive fetus. So the first time that they're exposed to that an Rh negative person is exposed to that Rh positive blood, they'll have a typical transfusion reaction. It's nothing out of the ordinary. It's that second transfusion will cause a reaction. You'll get a really good reaction. So I know we talked about this with some people on Tuesday. Hemolytic disease of newborn. It's also called erythroblastosis fetalis. And it occurs when mom is Rh negative and the fetus is Rh positive. The first pregnancy, mom is exposed to that Rh positive blood during delivery. The first baby is born fine, they're healthy, but mom makes those anti-Rh antibodies. Mom gets pregnant again with an Rh positive baby. Mom's anti-Rh antibodies start to cross the placenta and destroys the red blood cells of that Rh positive baby. They have to treat the baby with pre-birth transfusions and exchange transfusions after birth. We get it Rh-OGAM serum containing anti-Rh can prevent the Rh negative mother from becoming sensitized. So now this is given to mothers now during their first pregnancy. If they know one parent is positive blood type, one parent is negative blood type, they start looking at the baby to see, okay, what kind of blood type does that baby have? Do we need to start treating the mom? Yes, if mom and dad are both the same blood type, positive, then this isn't needed. This is only needed if, say, mom is negative, dad is positive, is when something like this is looked into. So we also have transfusion reactions, which can occur if mismatched blood is infused. So giving an Rh positive person an Rh negative blood, or even the wrong blood type completely out of the ABO blood group. So the donor cells are they're attacked by the recipient's plasma agglutinin. So they start attacking those red blood cells. It causes small blood vessels to get clogged, rupture. They release hemoglobin into the bloodstream. There's not enough oxygen carried through the blood when this happens. When we get a transfusion reaction, we get decreased blood flow because blood vessels get blocked. And hemoglobin in the kidney tubules actually lead to kidney failure. Patients usually get fevers, chills, a significant drop in blood pressure, rapid heart rate, they get nauseous, they start throwing up. So patients are treated with fluids and diuretics to help try to wash out that hemoglobin. So again, mentioning again, type O is a universal donor. So because there's no anti-A or anti-B antibodies on type O blood, so they're not going to cause any problems no matter who gets that blood. It's misleading as other anti-glucogens can cause transfusion reactions. So there's other things that must be considered besides the ABO blood types and the Rh positive, Rh negative. Some people are more sensitive to some of those other agglutinogens, and sometimes you don't know that until after they've received their first transfusion. So autologous transfusions is when patients predominates own blood and is stored and available when needed. So some people like to store their own blood for later use. That's kind of what this describes. So we did the blood typing on Tuesday. So whenever blood is donated, they always blood type it. They always look for the antibodies, the AB antibodies, because O doesn't have any. And then they also look for the Rh positive, Rh negative. So cross-matching and typing between specific donors with specific recipients. So they'll mix the recipient's serum with the donor's red blood cells. This is kind of how they cross-match to make sure that blood's not going to react before they put it in their system. They also mix recipient's red blood cells with the donor's serum. So they take red blood cells from the donor, put it in the serum of the recipient, and take red blood cells of the recipient and put it in the donor's serum and check for reactions. And this is what we had in our lab book on Tuesday and what we looked at on Tuesday. The AB blood type reacts to both A and B, the anti-A and the anti-B. Type A reacts to the anti-A. Type B reacts to the anti-B, and O reacts to nothing. So knowing that whatever it reacts to is what blood type it is I'm sure we've all had blood tests done, but they can give all kinds of information. They can be used to look at different kinds of anemia, blood glucose for diabetes, leukocytosis. The nice thing now is there's computers that you can put the blood in and it spits out the information. So there's not people sitting under a microscope. They may look under a microscope to get a better idea of what's going on, but the computer will kind of spit out how many red blood cells, how many white blood cells. It'll spit out usually the size of the blood cells, the shape of the blood cells, the color of the blood cells. That's all kind of done without somebody sitting there having to count each individual thing out. So the common tests done, actually a CBC, if you do a CBC with diff is what it's usually called, that includes this and this in the same test. So it'll look at the different types of white blood cells and what portion of each white blood cell is there, along with the formed elements. So you look at the red blood cells, the platelets, hematocrit, hemoglobin, that's all done. They can do a prothrombin time and platelet count. to look at hemostasis, clotting. They also can do a comprehensive medical panel where they check all the blood chemistry for levels that affect everything, including liver disease, kidney disease. They look for it all in one main blood test. Let's quickly look at the developmental aspects of blood, and this is actually our last slide for the evening. So fetal blood cells form in the yolk sac, the liver, and the spleen. Red marrow is primarily where those formed elements come from. And that usually happens by about the 7th month. So blood cells develop from those mesenchymal cells in blood islands. The fetus forms hemoglobin F. F is for fetus. They have a much higher affinity for oxygen than hemoglobin A, which is formed after birth. So hemoglobin F is for when the fetus is still in the womb and needs to needs more oxygen to develop. And after the baby's born, they create hemoglobin A to help, which is what we all have now, is that hemoglobin A to transport the oxygen around the body. So there's different blood diseases of aging. Of course, there's the chronic leukemias, the anemias, clotting disorders that are usually checked more often as you get older. And they're usually, a lot of times, are precipitated by or caused by disorders of the heart, blood vessels, or the immune system. So there's a lot of things that can cause blood problems. So there's a lot of different places to kind of look. That is actually it for today.